Good morning, everyone. We'll get started now. I'm Margaret Lynn, Director of the Clinical Research Development at the Tennessee Clinical and Translational Science Institute at UTHSC, and I, along with Dorita Brand, the Tennessee CTSI Director of Administration, want to welcome you to Research 103. The goal of this six-part series is to provide advanced information for research study conduct. And Research 103 follows 101 and 102, which were offered in the spring and fall of 2020. If you weren't able to attend those sessions, those recordings are available on our website, and you can go to the search engine of the UTHSC website and just enter Research 101 or Research 102 to find them. Alternatively, you can always email me at that says in Margaret, M. Lynn, L-Y-N-N, at uthsc.edu. We're going to record today's session, and all of the 103 sessions will be available on our website also. I want to thank UTHSC Information Technology Department for all of their help with this series. Thank you, Xavier. He's co-piloting. During our Zoom lectures, your microphone has been muted, but you'll be able to use the question and answer function to ask questions, and those will be asked at the end of the session. If there's any kind of technical difficulty during the session and we can't resolve it quickly, then we will post the session on our website and we'll send out an announcement of availability. So please just bear with us as we navigate through remote delivery of these sessions. You'll receive an evaluation of this session by email. After you complete that evaluation, you'll be able to download a certificate of attendance. So be looking for that evaluation within 10 business days. Dorita Brand and I have no real or apparent financial disclosures. So we'll get to the good part. Today's session is Protocol and Study Measures Training, the Importance of Accuracy and Consistency. And our speaker is Misty Thompson, PhD, CCRP. And Misty has a PhD in pharmacology. She has a basic science and clinical research experience through her graduate degree and 2 fellowships. She's experienced in both animal and human subject pharmaceutical studies. She worked at the UTHSC IRB as a compliance advisor. Many of you know her from that position, and we all love her. And she's currently employed with Penumbra. It's a medical device company, innovative medical device company, I might add. And she is a clinical research associate or monitor. So Misty, we're so thankful that you're here today to talk to us, and I'll turn it over to you.
Thank you, Margaret. Yeah, I'm happy to be here. Happy to speak to you all today. I just wanted to quickly go over a disclosure slide with you. So I have no conflicts of interest to disclose. I don't have any commercial support to disclose, nor any off-label product use to disclose. So as Margaret said, we're going to talk about protocols today and why it's important to be accurate and to be consistent when you're following your protocol. And we're also going to talk about what happens when you have changes either with institutional policies or just operational changes and how that can impact the study protocol as well. So we all know what a protocol is. We've all seen them. Some of us may have even written a protocol. So it is a formal document that describes our objectives, the study design, methodology, and statistical considerations, and just other aspects related to the study. The protocol serves several purposes. First and foremost, it ensures safety of human subjects when we're doing research, which is the most important thing to consider when you're designing your protocols and also during execution. It also standardizes the way that we collect data. So we want to make sure that the same data is collected in the same way for all of our subjects, because at the end of our study, we're trying to get information from this. We have, our objective and what we're trying to, a question that we're actually trying to answer. And you can't answer that question if you don't have data that is collected in a consistent way. And this is especially important when you have multiple sites in a study. So for sponsored trials, they don't want every site doing their own thing. They want everybody following the same protocol. And if you have a study, a PI initiated study, and you have multiple personnel on that study, you don't want everybody doing something different. So you want everybody doing the same thing. And it also feeds into scientific integrity, because we are basically saying, when we put forth this protocol, that this is what we're going to do and this is how we're going to do it. And ultimately, you are probably trying to publish this information, and you want to be able to say in that journal article that this is what we did and we followed this protocol. And, you know, the idea is that someone should be able to reproduce this and see, you know, similar effects were they to do the same study. And it kind of goes without saying, we don't have too many problems with this, but all study protocols that involve human research subjects must be IRB approved prior to implementing that protocol. And so I wanted to also give you some regulations and guidelines where you can find additional information after today's talk. So we're going to touch on ICH GCP. There's a section in there that discusses protocol components, and we're going to look at that on the next slide. I also wanted to provide you with some FDA regulations. So the two that I have listed here are the drug and device regulations. And so if you ever are submitting anything to the FDA, all the information that you need to know are in those two sections. And lastly, I just wanted to mention that if you are submitting an application to the NIH, they now have a template with instructions and sample text. And I've given you the link here if you want to go to that link, if you're interested in taking a look at that. This is for phase 2 or 3 clinical trials that require an IND or an IDE, and also for behavioral and social science studies that involve human subjects. So let's touch on the protocol components that I mentioned earlier. So all of these are taken directly from ICH GCP E6 Part 6. And so we could really give an entire lecture on just protocol components and how to write a protocol. But I just wanted to give you this information in case you find yourself needing to write a protocol and you don't know where to start or what should be included. I will note that some of these sections are geared more towards sponsored studies, such as, access to source data and documents. That's really for sponsored studies. But you can still take a lot of information from this. A lot of these are very basic, like background and objectives and what patient population you have. I mean, those things are going to be included with really any study, not just sponsored studies. And so let us think about this question. Do all studies require a written protocol? And so we've kind of just mentioned that if you're going to be submitting something to the FDA, like you have a treatment or you have a sponsored study, or you're going to be maybe applying for NIH funding, that you need a written protocol. But what if you don't have any of that as part of your study? So let's say you're just doing some data collection that's going to be banked in a repository, or maybe you're doing a retrospective record review or a straightforward survey study. Do you need a written protocol? So I will tell you, in my career, I've looked at a lot of IRB applications, especially since I've been working on the sponsor side. A good portion of them do require a separate written protocol when you go to, submit your study to the IRB. Some of them choose to put all of that information within the application. The one thing to think about is that IRB application will have questions that are based on that information that you would have in a protocol. So if you write up that protocol beforehand, it helps you think through your study design. It helps you think about feasibility, and also to kind of weed out any potential biases that you may have in your data collection, because those are things that the IRB reviewers will be asking when you submit that to the IRB, and they will be looking at those things. So if you kind of thought through it beforehand, then maybe you can kind of prevent a lot of those questions, or if they do ask the questions, then you know how you're going to handle that, and you already have thought about it. And so the other thing is that a written protocol can be distributed to all of your personnel, because again, we're trying to ensure consistency. So a lot of IRBs now have online submissions and everything is done online, but you can't just assume that everybody who's on your study are going to go into that system and download that application and know exactly what is in it. And I will say, once it's IRB approved, then you are required to follow exactly what you have in that application. So it's just going to help you ensure consistency in your data collection. And the other thing that I've seen happen a lot is a lot of these types of studies, you think they're really simple, but maybe you find something really, really interesting and you want to follow up on that. And a lot of these types of studies lay the groundwork for NIH grant submissions, or it may be something that gets incorporated into an FDA application later. So you can't just assume that it's never going to be part of any of those applications just because you don't have plans for it right now. And it doesn't have to be super elaborate. So I think a lot of times when people think about a protocol, you think about, you know, these sponsored studies that have hundreds and hundreds of pages in their protocol, and you may not have that. It may be very simple. It may be just even a couple of pages. So if we go back and look at those protocol components that we looked at, all of these that are highlighted in red are things that even the most basic study would have. So I think it's just, again, it's best practice to just go through, think through it, and write all these things out. And then once you and everybody who's on the study are in agreement, this is how we're going to do it, then do your IRB submission. And so feasibility is a big thing. So just because you have your protocol and you have it IRB approved, it doesn't mean that it's always going to be feasible for you and your site. So if a sponsor approaches you and they say, here's the study, are you interested? You need to think about whether it's feasible for you. And then if you're writing up a protocol for your PI initiated studies, there are some things to consider as well. So you need to think about how many staff members and investigators you're going to need to actually execute that protocol. And I feel like this is one thing that is underestimated a lot initially. And what we tend to see is as studies go along, people tend to add more staff because they've underestimated how many people they need. And you should also think about whether procedures can be formed in one location. So this doesn't always happen. I mean, we live in the real world and sometimes, you know, you need to do different things in different locations. So I would say if you're writing up a study, kind of think about that. And if you need to, you know, have things done in more than one location, maybe don't try to squeeze it all into one visit because patients don't tend to like running around, and that might actually impact your enrollment. So it's just something to think about. And you also need to think about the target patient population. So whenever you're looking at a protocol, you need to think about, do I have enough patients with these inclusion/exclusion criteria to actually reach the enrollment number that the sponsor is asking? Or if you're doing a PI initiated study, you probably have done a power analysis and you kind of know how many you need for the study. So just think about whether you have access to the patient population. Competing studies can be an issue. So we have investigators that are highly specialized. They might specialize in one particular disease state or one area of research. And so they may have multiple studies that, you know, are all enrolling the same types of patients. So you need to think about, again, do you have enough patients for all of your studies? And if you have too many studies that are being conducted simultaneously, it kind of gets back into having enough staff members to cover all of those studies. And you should think about whether you have access to all the necessary equipment and resources. So if you don't, you may need to collaborate with somebody. And if you are going to do that collaboration, that person would need to be on your IRB application as well. And the sponsor would need to know so that we have all of that information on file. And ask yourself whether you have adequate storage facilities. So a lot of times the investigational product or the supplies or the specimens, they have to be stored long-term. They may even need to be stored, you know, at a certain temperature. So just kind of think about whether you have access to that storage space. And also consider the length of a study. So not just for the individual participants, how long are they going to need to commit to the study and be involved in the study, but also the overall length of the study, how long you're going to be enrolling and how long is it going to take you to get the data that you need. So if you've ever been a part of a sponsored study, you know that they have site qualifications. Sometimes it's an in-person visit. They may want to check out your facilities, or it may just be in the form of a questionnaire. And then they come to formally initiate you for the study. So this is a really good time to bring up any concerns that you may have about the protocol, or whether you think you're going to be able to actually execute it. I feel like, just in my experience, a lot of investigators and site staff feel apprehensive about expressing concerns or declining a study. I think they think maybe they won't be selected again. If we say we can't do it, you know, are they going to approach us again for a study? And I will tell you, site selection is more complicated than that. It's not just if you decline a study and you have a very good reason, like we don't have enough patients to enroll, they're not going to just write you off permanently. And the other thing is, as far as expressing your concerns, if you already have a relationship with that sponsor, most of them actually value your input, especially if you have concerns about safety or the way a study is designed. They actually value that, and they will take that back to usually the national PIs and other committee members that are overseeing that study. So don't be afraid to speak up. And so in the title of the talk, we kind of talked about precision and accuracy when following a protocol. And I feel like sometimes these are used interchangeably, but they're not really the same thing. So if you've ever taken a stats course, then you probably have seen these definitions before. Precision is measurement of the repeatability or consistency of a measurement, where accuracy is the closeness of the measurements to a specific value. So if we think about this in terms of a research protocol, and I will tell you up front, I didn't come up with this analogy. This is something you probably have seen if you've taken stats classes, but I think that we can use this to very nicely illustrate following a protocol and the difference between precision and accuracy. So in the analogy, the research protocol is the bullseye. So that is what we are targeting, what we're trying to achieve. So high accuracy, high precision is the ideal study. So in this scenario, the protocol has been followed for all subjects enrolled. All of the data and information has been collected the same way for all of our subjects. But that does not mean that the outcomes will be what you expected. It may not even be favorable. So how many, FDA regulated studies do we know for drugs and devices where they don't get approved because they were not effective, or maybe they were not safe? That's a lot of it. It happens a lot. But at least we know in this scenario, because we have followed that protocol for all of our patients, that we can at least make an inference from that data. We can draw conclusions that we feel confident about because we followed the protocol that we designed. So what about high accuracy, low precision? So in this case, the protocol was followed for most subjects. It looks like we have a few minor protocol deviations that have fallen, outside of the bullseye. This is more realistically what we see with most studies. We know that there are going to be unavoidable circumstances that just happen, when you're dealing with human research subjects. Somebody doesn't come in to their visit on time, or, something that you just can't avoid. And so we just kind of expect that. In this scenario, we still have data integrity. We still can draw some conclusions, and we can, see what results we have. So it's sort of just the normal part of research. But what about low accuracy, high precision? So in this case, the protocol was not followed for anybody, but they consistently made the same mistake across all subjects. So they're all of the data is all clustered together, but the protocol wasn't followed. So this may be a case of someone did not understand the protocol. Another example is like an equipment artifact, something that is wrong with the equipment, so it just consistently, made incorrect measurements for all of the subjects. And as far as data integrity, it may or may not be compromised. It really depends on the nature of the deviations. So something like, someone didn't understand the inclusion-exclusion criteria, that would be a major issue, because now you've enrolled an entirely different subject population than what you wrote in your protocol. Something like an equipment error, maybe major or minor, and sometimes it's easy to put in your notes what happened. So I'll give you a good example. So after daylight savings time, one of the things I noticed is that some of the equipment, didn't auto set to the new time. So you might have one machine that says one time, one machine that says something different. And so it makes it look like when you're just looking through the data that, they were out of order or something like that. But those things are easily, noted, and there's no, compromise to the data. You can still do the data analysis. So it just kind of depends on what it is. And finally, low accuracy and low precision is what we absolutely don't want. So this protocol is not followed for anybody, and it is not consistent across subjects. So it's just all over the place. So the first issue with this is that you couldn't make an inference from this data. You can't even know, you can't even answer your question, right? Because we haven't followed the protocol for anybody. The other thing is some of these would be considered major protocol deviations because the farther you get away from the protocol, the more you could have potentially harmed subjects depending on what it was. And so let's look at protocol deviations. Some people call them violations. And so your protocol may outline how deviations are going to be handled, or that may be something that you want to put in there if you're writing a PI-initiated study, to just say up front, if the following happened, we won't be using the data from those people. For example, if the inclusion and exclusion are not met, they won't be included. Things like that can kind of help you with any potential bias in the data, and just saying upfront what is going to be included and what is not going to be included. All deviations should be documented, and there are different ways that you can do that, but they should be documented in the research records so that if anybody, like a monitor or the FDA or anybody looked at your records, they would know what happened. There's also no universal definition for major versus minor, and that's something that we get asked a lot. This is usually determined by if you have a sponsor, they have a definition. Most IRBs have a definition. So just consult with them if you don't really know whether your deviation is major or minor. In general, some of the things that we look at is, could it have potentially caused harm to a subject? Does it involve non-compliance of a federal regulation? Did it compromise the data integrity? And one of the things I used to hear a lot is that, yeah, we have a lot of deviations, but they're all minor. The thing to consider is that if you have so many minor deviations that you can't even analyze the data, that sort of culminates in a major violation, right? Because now your data integrity is not there. So it's just something to think about. They may also be immediately reportable to the sponsor and the IRB. It depends on the nature of the violation, and also depends on their policy. So again, I encourage you to reach out to whoever you're working with and find out what their policies are on that so that you will be in compliance. Very rarely, they may be reportable to the FDA or the OHRP. It doesn't happen that often. It's usually something very serious, or like I said, there's so many violations that, you couldn't even really analyze the data, things like that. If a protocol deviation keeps popping up over and over, first of all, it may require retraining of your staff because they may just not understand what they're supposed to do. And for sponsored studies, we do a lot of retraining when there are protocol deviations. And I would encourage you to develop a corrective action plan. So we don't just want to know that you understood why it was a protocol deviation, but what are you going to do moving forward to prevent those deviations from happening? And the IRB may require that you draft this and submit it to them as well. And it may also mean that an amendment is needed. So for sponsored studies, if we see, that all sites are struggling with the same aspect of the protocol and there are tons of deviations, that may be when we step back and go, okay, is this feasible? Do we need to maybe make some changes to the amendments? So it's something that you may need to do as well with your PI-initiated studies, just to see whether you need to make an amendment to make that protocol a little more feasible. And so protocol amendments are really any change to any aspect of the study. It may be a minor change, like an administrative change. So for example, increasing the number of subjects or changing your clinic location. They may be, administrative, or it may be major. So examples would be changing your inclusion exclusion criteria, adding on extra visits, adding on extra, collection of specimens or extra procedures. Must be IRB approved prior to implementation. And that may seem obvious, but that's something that I saw a lot when I used to audit with the IRB, is that people would make changes to their protocols without, getting IRB approval for it. And people would say, well, what's the big deal if we don't have IRB approval? It may change the risk benefit ratio. And so what do I mean by that? So it may change, if it increases the risk or decreases the benefit, then it may change the approval categories. So for an example, if somebody is changing the age range of their subjects, and now you're going to enroll minors, that's a completely different category under the federal regulations, and that makes it a higher risk study. Or if you're adding, procedures that are, have more risk involved, that may change the way that they actually review it and the categories that they review it under. And if they approved it as an expedited study, if you're increasing the risk, they may now have to review it as a full board study. And the other thing to note is that if it is an FDA regulated product, you may also need to submit to the FDA prior to implementation. And so the other thing to point out is that if you do now have a new protocol, you want to ensure that all of your study staff and your investigators are trained to the new protocol, so that they're not just continuing to follow that old protocol, because now you're going to potentially rack up even more protocol deviations, which is what we were kind of trying to avoid. And so what about the consent form? So a lot of times when you have protocol changes, especially the major protocol changes, that also does require changes to the consent form language, which should also be IRB approved, and it should also be sponsor approved as well. So if you have to make any changes to your consent form after the sponsor has approved it for whatever reason, definitely route it back to the sponsor, because they have to approve that internally as well. And so the question is, should subjects be reconsented? And that's something that I got asked a lot. And I will say that sponsors and IRBs all have very specific policies about this. And so again, consult with your sponsor or your IRB to find out, whether you need to reconsent for a specific protocol. The general rule of thumb, however, is really, could it change their willingness to participate? And so things would be like modified procedures or changes to the study design. Those people are now going to have to consent to the new procedures that you're adding. If there are changes to the safety information or changes to confidentiality, if you read the consent form language, it actually says, if there are new information about this drug or device, we will let you know during the course of the study and afterwards as well. So that leads to the next question. What about people who've already completed the study? Do we have to reconsent those people? And the answer is, it kind of depends. So they should be notified of changes in the safety profile or changes to the way their information or specimens are being used. So a good example is, I've seen some drug studies that collect blood specimens or information as part of the drug study, but now they want to use it for a repository. But you can't just make that switch, use that information for a repository without their permission. So that would be a separate consent. It may be in the form of an addendum or a letter, depending on what it was. So if, for example, it's new safety information, you may be asked to send a letter to the former participants to let them know. Depending on what it was, they may need to come back in for some safety checkups, or they may want to go to their primary care physician and follow up, things like that. So it may not be necessarily a full consent form, it may just be a letter or an addendum to the consent. And so what about changes to study operations? Excuse me. So there are sometimes changes to site operations, the way you actually carry out a protocol, or institutional policies that may not necessarily result in a change in the study protocol, but because it changes the way you execute that protocol, you may need IRB or sponsor approval. And so I've given a lot of examples here. So if you update your site or staff, your investigators, or if the investigator status changes at the institution, the IRB and the sponsor will want to know about it. If you've changed the way you have your delegation log, people are, assigned a certain task. If they're going to be taking on a new role, you would need to let them know. Any updates to financial disclosures or changes to your site location or the way you're storing a product, things like that can change the way you execute a protocol. And then lastly, if there's any alteration to the consent process. So if you are changing the way you're doing the consent, and we're going to talk a little bit about this later in the talk, you need to let the IRB know. First of all, they have to approve it, but then the sponsor has to be okay with it as well. And so for the last part of the talk, I really wanted to talk a little bit about COVID and how it's impacted the clinical research landscape. We're all very acutely aware that things are very different now as a result of COVID. But what was really interesting from my perspective, working with the sponsor, is I got to work with, because I work with institutions that are all over the United States, I got to see how different institutions were handling this. And it definitely, I would say, impacted every single research protocol that exists. I would say there are very few, slim to none, that didn't get impacted at all. And so it's a good example of when, circumstances happen that are beyond your control that result in either, operational changes at the institutional level or may even result in amendments to the protocol. And so we were all faced with really, some very tough decisions. I wanted to provide you with an FDA guidance document. So this was first put out in, I believe, March of 2020, and they have just been amending it as we go along. The overall message, if you do go in and read that guidance document, is that there's no universal answer for every single study, every institution, and every patient. So you really have to evaluate it on a case-by-case basis and figure out, what is best in the situation. And so their overall guidance basically says, revise your study protocols or your institutional policies as necessary, with the goal of protecting patients first and foremost, maintaining your data integrity, and to minimize those protocol violations. So the first thing I think most people were faced with the decision about was enrollment. Do they continue to enroll? And if they do enroll, which studies are they going to enroll in? Some institutions did decide to suspend all of their studies at an institutional level. And the main reason was they wanted their employees to be able to focus on COVID and helping on the front line. And a lot of research staff did that. The downside is that patients had, less treatment options available because of it. Most of the institutions that I worked with chose to do, suspension of what they considered to be non-essential studies. There was not a universal, definition of essential versus non-essential. Each institution had their own policies on this. The advantage for this approach was that some of the drug and device studies got to continue to enroll. The downside was that a lot of the non-COVID PI initiated studies suffered because they were, labeled as non-essential during that time and they were suspended. And then some of the sponsors chose to suspend a study at all sites. Just kind of pause, everybody stop what you're doing, just to keep all sites on the same level. The downside was that sites felt like it might not be fair. So we had a lot of sites that weren't really impacted as much and they could continue to enroll. So they felt like it was not fair to stop enrollment, just because other sites couldn't enroll. The other issue with that is that you still have the cost of maintaining the study. Even if you're not enrolling, you still have IRB submissions going in. It also extends the length of the study, the overall length of the study. So you're going to have to do an amendment and those are going to have to go to the IRB. So it's not less work just because you're not enrolling. And for that reason, we kind of chose to keep all of our studies open and let each institution decide what was going to happen at the institutional level. Some sites chose not to limit enrollment in any capacity, but they still saw reductions in study participation. So we all know that There was a major reduction in patients going to the doctor. They weren't seeking treatment even for standard of care. So that meant that they weren't going to be enrolling in any studies either. There were also a lot of restrictions on which procedures could be conducted. So a lot of our sites, a lot of our institutions were not allowing for elective procedures. It was emergent procedures only. And so some of our studies suffered for that reason. A lot of patients didn't come in for their routine visits. So again, it's kind of hard to capture those patients and get them involved in research if they're not coming to the clinic or the hospital. There were some studies that were close to enrollment before COVID. And so, what is common in a lot of research studies is they will do interim data analysis. So every once in a while, we'll kind of look at the data, see the trend. And even before COVID, you would see, some studies closing prematurely if they felt like, it wasn't going to make any difference, or they were really close to enrollment and they were just not seeing the results they were looking for. A lot of times they closed for futility reasons. So we did have some people, some studies closed early because of that. And so obtaining consent during COVID times also very challenging. What we saw is that a lot of staff were not allowed on site. Or they had restrictions on where they could go in the hospital. They weren't allowed to go into the patient hospital room. So we saw a lot of people adding on e-consent, which is electronic consent, or doing like a telephone consent or a use of a video call. These things can be very, very useful, especially during current times. The thing I will say is that sponsors and IRBs have very specific policies about how this should be executed. So you can't just start doing it, you realize, oh, I can't go in this person's room, and you just execute a telephone consent. I used to see that a lot when I did IRB audits. So you definitely need to write that into your protocol, your IRB application, and get that approved. Some sponsors don't, most of them do allow for these types of things, but some of them may have very specific guidelines on how they want it done. The thing to note is that federal regulations still apply. So you're still going to have the same consent information. The same information has to be presented, the same, ways that you would do that, making sure they have, a copy of it, and making sure that they have all the information that they need to make a decision. All of that is still there. It also may require, Part 11 compliant systems. And so this is the section of the FDA regulation that basically says, these are the requirements if you're going to be doing, electronic systems and electronic signatures. And so sponsors or sponsors or IRBs may require that you present something that shows that that system that you're using is Part 11 compliant. And so the other thing that got really messed up during COVID is treatments and interventions. So the question is, do we continue these treatments? And if so, which ones do we continue? Do we change how we're administrating our study operations on a site level? And so again, many patients didn't feel comfortable going to the clinic or the hospital. So they may have already been enrolled. What if they were already enrolled in a study and they don't feel comfortable coming in for their treatment? So one option is to discontinue treatment or intervention. The question is that safe? For some, investigational products, it is not safe to just stop them. They may need to be worked down on the dose, or if it is stopped, they may need safety visits for a while after that to make sure everything's okay. One of the things that really determined for a lot of institutions whether a study was essential or non-essential is whether or not they could get that treatment outside of the study. And so for us, we had a couple of device studies where it was a post-market study. So the device was already FDA approved. The patients could get it outside of the study. So some institutions would not allow our sites to enroll for that reason. Most of the sites chose to continue treatment or intervention, but then the question is, how do they receive the product? So there's some logistics here. Can they still come on site and pick up the product, or do we need to maybe deliver it to their home or another location? And with delivery, it gets tricky because you have to think about who's going to receive that product when it gets there. And if it's not FDA approved, I mean, it's kind of dangerous depending on what it is. And then you have compliance with their drug or device. How are you going to know that they're using it if they're not coming in, to get it? And the other thing is destruction. A lot of those drug studies, they expect you to destroy that drug. And if they're, if it's being sent to their home, how do you know that they're destroying it? Or I'm sure there would need to be some sort of information for them on how they need to actually do that. So it's just a lot to think about if you're going to take that route. I would say that the treatment studies that require on-site administration probably had the most issues. So if it's like an infusion or a device that is not portable, things like that, may require you to collaborate with another site who maybe doesn't have as many restrictions. But then again, that would need to be IRB approved, as well as any other changes to how you're administering or utilizing, those products that would require a protocol amendment, and it would require, IRB and sponsor approval as well. And so aside from receiving treatment, what about just regular follow-up visits that we have written in our protocol, or other study procedures that we have written in there? Most of the institutions advised their staff to conduct telehealth or telephone visits whenever possible. Sometimes it's not possible. So things like labs and diagnostics and physical exams, other measurements, that's more challenging to do from a telephone visit. And so again, we saw a lot of people collaborating with other centers to try to work around and actually make it feasible for their patients to still have their follow-up visits. And again, would require an amendment and IRB approval. And so I wanted to touch on monitoring because it's something that, I had to do or do on a daily basis, but most institutions did not allow outside visitors, which is understandable. A lot of sponsors suspended travel. We did because we saw ourselves as super spreaders. We're going in and out of hospitals all over the country. And so we didn't feel like it was safe to our institutions or to the patients at those institutions for us to be going, in and out of their hospital during that time. And as you all know, if you've had to maybe deal with remote monitoring from a site standpoint, you know that it's very challenging. So a lot of times, the IT departments don't allow access to medical records, which is understandable. I mean, we're outsiders, we're not part of the institution. And so a lot of times it was just explaining to them why we need access. And then some of the site staff weren't on site, so they didn't have access to the records. They didn't have access to the study records or the medical records. And so it was just challenging for everybody. And the other issue is if you don't have access to those medical records and they're on site, that requires them to do a lot of scanning and sending, and it's just a lot of work. A lot of monitoring visits were just postponed or done out of window according to our monitoring plan. We have a monitoring plan which says this is how frequent we're going to monitor the data depending on what type of study it is. So we weren't always able to adhere to that. A lot of sponsors are now going to risk-based monitoring. And so that means different things for different companies. So it's usually a type of monitoring where, depending on the risk level of the study, they will choose to monitor absolutely everything because it's high risk, or they'll just pick certain data points or a certain percentage of the data and just look at that. And what we did notice is that as time went on, I think a lot of IT departments started understanding that, this wasn't a temporary thing, and maybe they need to, to reevaluate their policies and allowing people to access those records remotely. And so I had to do things like get background checks for some institutions, and they basically treated me like a new employee, and that was fine with me. And I will tell you, most sponsors are going to be amenable to doing those things because they need to monitor the records. So they're going to be willing to do what they need to do to get access to them. And so the FDA has basically said with regards to data and documentation that they are expecting more protocol violations during this time. They already know there are issues. They're expecting, more variability in the data. If you have missed visits or incomplete data due to the pandemic, you should be documenting that in the research records the way you would anything else that happens during the course of the study. We may need you to change your methods or implement amendments And if you do that, then you should document that in your research records. Why, the first 5 patients were following this protocol, and then this happened, and we changed our amendment, and this was the date that it was approved, and then the rest of the people were on this other protocol. You may need to revise your statistical plan or how you're going to analyze the data to account for, that missing data. And so what we're hoping to see, or I think what the FDA is sort of hoping as well, is that people will do the amendments that they need to do and change their operations so that they're, seeing more of the target that is down at the bottom, more minor violations, and not, so much data that's just all over the place because of the pandemic. And so the last thing I wanted to leave you with is sort of a research outlook. So we know things are getting better, but things are not perfect. And what is the, research landscape going to look like moving forward? I think personally, we may be looking at a new normal. Before COVID, there was already a shift towards more electronic records and less paper, because a lot of archives are looking like this cartoon down at the bottom, and they just have boxes and boxes and boxes of paper records. And for that reason, they were already kind of shifting towards electronic. We're seeing more sites do e-consent and e-regulatory and just using electronic record systems. They're also becoming more secure. So I think a lot of companies are recognizing that there's a need for this now more so than ever. And so they're making their systems more secure. More systems are offering Part 11 compliant options. So Adobe now has a Part 11 compliant option that you can purchase. DocuSign, again, you can purchase a Part 11 compliant version of it. I think that monitoring may be more of a hybrid of on-site and remote. And the more we shift toward electronic systems and electronic records, it's going to make it easier for us to do that, and less work for the site and less work for the monitors when things are electronic. I think that we will see, more telephone and telehealth visits being incorporated into the protocol routinely. We already were doing a lot of that before, but I think we're going to see even more of it. And lastly, I think protocols will be designed with more flexibility. So rather than having very rigid, visit windows, for example, and very rigid, methods, I think we'll see like more options for the patients, or, visit windows that are, a little more realistic. I think we'll see that with protocol design. Yeah, and that's it for me. Margaret, do we have questions?
Yeah, we do. Misty, that was an amazing presentation. Thank you. And there are a few questions.
Okay.
The first one is regarding an LOA, and I'm guessing that this is a letter of agreement preceding an amendment. Is it a valid process to obtain ICF with an LOA?
Yeah, I mean, so assuming that that's letter of agreement, so I guess they're saying with the sponsor, is that, I'm guessing, correct?
It wasn't clarified, but
okay. Yeah, well, I know
with the CRO, Misty, it could be with the CRO also.
Oh yeah, that's true. No, I mean, so I think what they're saying is that if they have an agreement that this consent form is going to be used, I mean, you would still need IRB approval before you implement it, no matter
what. Okay. The next question, as a monitor, what do you find are the major accuracy issues you see when you visit sites? If you give a few.
Issues. Mm-hmm. Yeah, I think that it kind of goes back to what I was saying. They don't want to speak up and say when they have issues with the protocol. So I see, investigators, especially if they have a standard process that they do, a standard of care process, a lot of them will revert back to their standard of care versus following what is in the protocol, because that's just kind of what they do day to day. And especially if they may not like that particular part of the protocol, they'll say, well, this is what we do, and this is how we did it, and it's like, those are the types of things that you would need to bring up again, when you're doing your site quals and your site initiation. And if this is not the way you normally do it, is not what is in the protocol, and you're not comfortable following that protocol, you would want to bring that up, because we see that happen a lot. And I think as far as, study visits, that's a sticky point for a lot of people, especially those protocols that have very, narrow visit windows. We see a lot of out of window visits. For us, my company handles those a little bit different than some sponsors do. I think probably a lot of people get tons of protocol violations for that, or things like lab draws, or, things that are just maybe not feasible at the time during that study visit. Those are things that are just very common.
Okay. What do you think, Misty, is the best method to train staff on accuracy?
Yeah, so I think it's great whenever we're doing an SIV, or, if you all don't have a sponsor, you can kind of do your own SIV. That would be great, where you just get everybody in the same room at the same time and go over the protocol in detail and just make sure that everybody's on board, as we don't see a lot of that happening. I mean, when I was an IRB auditor, I didn't see that a lot of training happened on the front end. It was more like wait until there's an issue and then let's go back and train people. I think if you do more training on the front end and get everybody on board, at least the PI and the coordinators, if you can get all the investigators in the same room at the same time, it's really hard to do that, but I think it really helps. And what we also see on the sponsor side is sometimes even people who are not going to be involved in the research protocol specifically, maybe like lab staff, or if you have people who are going to be in the OR, even if they're not going to be involved in the research procedures, just getting them involved so that they know that the study is going to be going on, right? And they know that, okay, things may not be done the same way for this person because we're not following a standard of care for this person, and just keeping them involved as well, I think is a good idea.
So would you require any kind of documentation for competencies on whatever procedure is being done for data collection?
So for the people who are on the protocol, on the research, or people who are not on the protocol?
For those that are on the protocol as part of the key study staff?
Yeah, so we usually, we collect medical license and CV as like a routine thing, but then during the site quals, they tend to ask a lot of questions about the investigators, like how many years they've been doing this procedure, how often do they use this product, or if it's not an FDA product, similar products, things that are already, FDA approved, and they get an idea of like kind of what their experience level is through that.
Next question. What would be the tipping or triggering points to shut a study down from the sponsor's perspective? You as a monitor go in, and what's going to cause that to happen?
Yeah, so usually their first step, you're going to get a lot of warnings. It's not like it's going to be a secret. So whenever we come for a monitoring visit, whatever we find is going to be in your monitoring letters. And then we're going to follow up on that, at the next monitoring visit. And if it just keeps accumulating, the same protocol deviations or things that are outstanding that you're just not taking care of, we ask you to do something repeatedly and it's not getting done. Also, if a PI is not involved and there's no documentation of PI involvement and things are getting worse and worse and worse, eventually they're going to suspend you, and it wouldn't be a permanent close. It's usually just like, we are suspending you until the following can be sort of taken care of. And usually at that point, we would require, a corrective action plan. We would ask that person, contact your IRB and see how much of this is actually reportable. And we usually ask for IRB protocols, because every IRB is very different in terms of what they want reported and how they want things handled. And so we usually get copies of IRB protocols for everybody that we work with. So.
Okay. Dorita, do you have any questions?
No, ma'am. I think they were all answered. Misty, thank you so much for presenting for us today. As always, you're a wealth of knowledge for us as researchers, and your presentation, as a couple people have said, was excellent and gave us so much good information that we all, no matter what level of experience we have, can use when we're conducting our research projects. We really appreciate you doing this for us.
Yeah, no, I enjoy it. And I always say, like, I'm an open book. If people want to contact me afterwards and just ask me questions that they didn't want to ask in a group setting, they're welcome to do that. I don't know, Margaret, if you're providing contact information, but you're welcome to do that.
Anybody can contact me at mlin@uthsc.edu. Sure. Mm-hmm.
Well, we thank everyone for attending today's session and hope you join us for our next session, which is this Thursday, the 22nd. And Dr. Mona Wicks will be presenting the topic diversity and inclusion in research. And we thank everyone again, and especially Misty and Margaret, and I wish everyone to have a good rest of your day.
Bye.
Bye. Thanks.